This is the second part of the lipids, membranes, and membrane transport. And in this lecture, we're going to discuss more about the membrane proteins as well as the membrane transport systems. Membrane proteins are the proteins that are present in various membrane systems of the cells, such as plasma membrane, nuclear envelope, as well as organelle boundaries. And what you're looking at here is an integral membrane protein which spans across the lipid bilayer of the membrane. And these integral proteins, as well as other membrane proteins, contain hydrophobic amino acid side chains oriented outward on their interface for basically interacting with the nonpolar phase within the membranes, such as in these phospholipids. And since they contain the hydrophobic amino acids, and they're generally insoluble in aqueous solutions such as water, and therefore they have fewer hydrophilic amino acids than cytosolic proteins. Membrane proteins have several types, and the first one that we'll discuss is integral proteins, which are basically embedded inside or in the middle of this membrane lipid bilayer. So they are tightly linked to the membranes, and they can be separated from the membrane only by the treatment with agents that can disrupt the membranes, such as detergents. And they associate with the membrane through strong hydrophobic interactions through the hydrophobic amino acid side chains that are located in the mid region of these proteins. And they tend to aggregate when extracted into an aqueous solutions such as water because of the hydrophobic effect that we already discussed in the water section. And the domain that is present inside of these integral proteins is rich in alpha helix and beta sheet structures, and removal of integral proteins from the membrane tend to disrupt the integrity and the structure of the membrane. So they definitely have a role in the forming and keeping this membrane structure. And examples are the glycophorin A in human erythrocytes and porin in bacterial cells. And some of them, and quite many of them, actually have this kind of pore or the channel-like structure, which will have a critical role in exchanging some substance inside and outside of this organelle or the cell that is encapsulated by the membrane. Another type of membrane proteins is peripheral proteins, which can be dissociated from membranes by simple treatments such as high ionic strength of the solution or by change in pH. The peripheral membrane proteins are basically just touching and interacting partially with this lipid structure, so the interaction with the membrane is relatively weaker than the integral proteins. So basically it can be pretty easily dissociated. And generally these peripheral proteins are linked to other proteins, particularly integral proteins. So there are going to be some protein-protein interactions between integral protein and peripheral protein using some hydrophobic interactions and such. And these peripheral proteins are soluble in an aqueous solution used for extraction. So there can be some optimization needed for such making these peripheral proteins dissolved in the aqueous solution, such as certain concentration of the salt, but they're readily soluble. And the removal of peripheral proteins leaves the membrane intact, meaning that again they're just interacting partially or in the just one side of this protein with the membrane. So even if these peripheral proteins are removed, they're not going to have a huge effect on the integrity of the membranes. And the examples for such peripheral proteins are cytochrome C, which is involved in electron transport chain during oxidative phosphorylation, which is a critical process for the ATP generation or the energy production and storage in the cell. And we're going to discuss much more about that in the later of this course. And another one is lipid-linked proteins, which proteins are covalently linked to the lipids that anchor them to the membranes. So instead of these peripheral proteins that are interacting directly with the lipids partially or to the integral proteins, the lipid anchor proteins uses these lipid anchors to be held and stuck in the membrane. And a single protein may contain more than one lipid group linked, and generally they are present on the inner lipids of the lipid bilayers. An example would be the attachment of the myristic acid, which is a lipid acid, fatty acid, to make the myristylation. And the palmitic acid is another widely used the anchor, which make the palmitylation on the surface of some proteins. And also there are glycoproteins, in which case the proteins are linked to the carbohydrate moieties instead of lipids, and they are generally present on the outer lipids of the bilayers for cell signaling or some other cell biological processes. Among lipid-anchored membrane proteins, there are 4 subtypes. The first ones are mild-linked myristoyl anchors, which structure you can see right here. And the process of anchoring between this protein and the lipid will be called as N-myristylation. And the next one is a diester-linked palmitoyl anchors, which you can see the linkage here, and it's called as palmitylation. And both of them are basically fatty acylated proteins. The third one is a thioester-linked prenyl anchors, where you can see these 2 examples. And prenylated proteins are the proteins covalently attached hydrophobic isopren polymers as cysteine residues, especially of these proteins. And these isoprenoids groups are usually the farnesyl, which is 15 carbon, or the geranyl, which is 20 carbon. And the last one is glycosyl phosphatidylinositol anchors, or GPI anchors. And this is basically the protein that are attached to a GPI complex molecular group beyond amide linkage to the protein's C-terminal carboxyl group. And on the membranes along the lipid bilayers, all molecules, including the lipids, move continuously by simple diffusion. And heat energy is involved there to cause these molecules to move randomly, such as Brownian motion, and thermodynamics is involved in such processes. And if the concentration of molecules is different in 2 regions, so it builds up on a concentration gradient, then diffusion will cause the molecules to move from a region of high concentration to one of low concentration. And the higher the concentration gradient, the more rapid the net diffusion. So it won't affect the speed of each molecule's movement, but it definitely will accelerate the time that this concentration gradient is dissipated. And diffusion evens out the concentrations so they are equal everywhere to maximize entropy. So the membrane fluxes, including the lipids as well as the proteins, are driven by forces existing across the cell membranes. So such flow or flux of the material is proportional to the gradient of a force acting on the molecules. So the bigger the gradient, the larger or stronger the force will be. And the forces arise because conditions are different on 2 sides of the membrane or 2 different regions of the membrane. So this table lists a different type of fluxes as well as their conditions and the forces that is driving such flux. So the first one is a diffusional flux, which was driven by the concentration differences on 2 different sides or the different regions in the membrane, and that concentration differences build up a concentration gradient to induce such flux. And the second type of flux is electrical current, which is due to the difference in voltage. This difference builds up a gradient also in voltage too. And the third one is bulk flow in volume, and that is due to the difference in pressure, which builds up a gradient in pressure. And the last one is osmosis flow, also work in the volume. Is due to the difference in osmotic pressure in two different sides of the membrane, and that builds up again the osmotic pressure gradient to drive such flow. And the flow or flux of materials across membrane by simple diffusion is called the permeability. And this permeability is a spontaneous process that does not require energy input from the ATP or any other sources. And the flux is proportional to the concentration gradient because it also is the simple diffusion. And no saturation has been seen there in such diffusion. And the flux is always downhill, meaning that it just follows this concentration gradient from high to low. And hydrophobic substances have a high permeability through bilayer membranes. That means it takes a shorter time, or these chemicals move across the membrane faster than the hydrophilic chemicals. The first stage of such flow across a membrane is a partition from the water into the membrane. And hydrophobic molecules partition better into the lipid membrane because of these hydrophobic interactions between the lipids on the membrane and the hydrophobic molecules. And many biological chemicals are deliberately made hydrophobic to increase in their rate of penetration into cells, such as many drugs, as well as pesticides like DDT. And osmosis is the process that moves water across biological membranes and causes volume changes in the cell. It is useful to think that a dilute solution as having a high water concentration and a concentrated solution as having a lower water concentration. This is not true because the water concentration is all the same everywhere, but it's useful to think this way to better understand how osmosis works. If we think that this way, then the water flow goes from the high water, which is from the dilute solution, to low water concentration, which is a concentrated solution. But actually, the more accurate picture will be the solutes in lower the free energy of the water. So you have more solutes in concentrated solutions, so you're going to have less free energy of water, as opposed to a dilute solution will have relatively high free energy of water, so it can move freely to the other side where there are more concentrated region is. The cells basically swell in hypotonic solutions and shrink in hypertonic ones. And cells have developed different ways to combating these osmosis because otherwise, if the cells cannot control it, then it will simply burst. So the plants, fungal, or bacterial cells developed in a way that they make the cell walls around the plasma membrane that are really rigid, and they can prevent from swelling because the walls are strong enough to allow the fairly high pressure gradient. And most cells, especially animal cells, pump ions out of the cell, which reduces the internal osmotic pressure. So you're going to have less ions inside of the cells, which lowers the concentration of the cytoplasm. Then it'll reduce the osmotic pressure, so prevent it from bursting. And the most important pump among these pump ions is the sodium-potassium pump that we're going to discuss more in depth in a few slides. Passive-mediated transport, also known as facilitated transport, is the process of spontaneous passive transport of molecules or ions across a biological membrane via special proteins or specific transmembrane proteins that help move substances across membrane. And they are typically very specific, which allows cells to select substances that they take up. And they are sensitive to inhibitors that react with the protein side chains. So you can use drugs, or there are genetically coded proteins that works as an inhibitor for such proteins. And ATP energy is not required for the passive mediated transport, again, because this is a spontaneous passive transport method. And the transport rate reaches a maximum when all the protein transporters on the membrane are being used, so they're going to be in saturated condition. But it cannot transport molecules against a concentration gradient, meaning that there are not going to be any uphill transport in terms of energy or the concentration. Membrane transport proteins are typically multipass transmembrane proteins, so they can have these secondary structures such as alpha helices or the beta sheets. And the winding of the polypeptide chain within the lipid bilayer allows hydrophilic solutes to pass through the membrane without having near contact with the hydrophobic interior of the bilayer. And there are two main types of membrane transport proteins, carrier proteins and channel proteins. Carrier proteins bind to the solute and undergo a conformational change to transport the solute across the membrane. But channel proteins form aqueous pores, allowing specific solutes to pass through or across the membrane. And these channel proteins allow much faster transport than carrier proteins in general. Transport through carrier proteins may be either passive or active. And here, passive transport, meaning that it's basically a spontaneous process, as opposed to active transport, meaning that it requires an input of energy. But the transport through channel proteins is always passive. In other words, solutes pass through the membrane in the downhill direction all the time of their concentration gradient, but it never goes uphill. In active transport, solutes are pumped across the membrane against their electrochemical gradient, and this requires the expenditure or the input of energy, usually via hydrolysis of ATP, but it can be different form of energy too, such as heat or light or the electricity too. But in the passive transport, the electrochemical gradient is the effective driving force across the membrane for a given solute and is a function of both the concentration gradient and electrostatic membrane potential too. For active transport, there are three main mechanisms: coupled carriers, ATP-driven pump, and light-driven pump. So for the coupled carriers, a solute is driven uphill. So uphill is going to be from low concentration to high concentration. So this molecule moves here, but that will be compensated by a different solute being transported downhill. So for this red rectangle molecule, they're going to be the concentration gradient setup, where this is high concentration for red rectangles, and this is low concentration. So it'll be a natural downhill transport for this type of substances. So the second one is the ATP-driven pump, where the uphill transport is powered by ATP hydrolysis. And the last one is a light-driven pump, and this is basically the uphill transport by the energy input from the photons. And an example would be the bacterial rhabdosin. Unlike a uniporter, which handles just one molecule at a time, coupled carriers move two molecules at the same time, and there are two subtypes for coupled carriers: symporters and antiporters. And symporters are basically the one that transport the second solute too in the same direction, both of them at the same direction. But the antiporters are the one that the second solute moves the opposite direction. In the plasma membrane of animal cells, the second solute for the coupled carriers is often sodium ion, with a concentration much higher in the extracellular space compared to the cytosol. And an example you're looking at here as a coupled carrier is the glucose carrier, where it consumes the sodium ion as well as glucose again to push into the inside of the cell. So it's a symporter. And the gradient of this sodium ion, which drives the coupled carriers here, is itself created by the ATP-driven transport of sodium ions. So that means somewhere in the plasma membrane here, there are the sodium ion transport, which consumes the ATP to push out the sodium ions to the extracellular space. Therefore, the coupled carrier transport is termed secondary active transport because it indirectly uses the ATP there. So it's an active transport when you look at it in a big picture, while the ATP-driven transport is termed the primary active transport. And in coupled carriers, the binding of the two solutes to the transmembrane protein is cooperative. So changing from state A to state B, all of them are happening at the same time in cooperation. One of well-known coupled carriers is a sodium-potassium pump, which maintains the sodium ion gradient across the cell membrane, and it is also called as sodium-potassium ATPase because it consumes ATP for such processes. And this is an antiporter, meaning that it pumps out the sodium ions out of the cell and importing the potassium ions, both against their electrochemical gradients. And you can see its cycle of the processes here. And basically what it does is pump out the 3 sodium ions and also import the 2 potassium ions per cycle. And this pump is powered by ATP, and that's why it's called as ATPase. And strikingly, about 1/3 of ATP in an animal cell is used to power these pumps. So it consumes a lot of energy to keep this gradient. And electrically active nerve cells, which use sodium and potassium ion gradients to propagate electrical signals, use up to 2/3 of the ATP for powering such pumps. So again, the sodium-potassium pump is to maintain the electrochemical gradient of sodium ions as well as potassium ions by consuming the ATP to push out the 3 sodium ions and importing 2 potassium ions. But this pump can also be driven in reverse, with the ions moving down their electrochemical gradients, so moving in opposite ways for both ions. That is to produce ATP from ADP and phosphate. And if this pump is inhibited, so it's not working, then cells will swell up and burst. So far, we talked about the active transporters, which consume ATP as well as some other energy sources. Now let's turn to ion channels and their structures and mechanisms. These channel proteins allow passive diffusion of solutes. And some ion channels are large and less specific, such as porins, and those channels help form the communication channels between cells typically, and they are called gap junctions. But most channel proteins are highly specific and can have either open or closed configurations. And channel proteins, when they're in open configurations, process 10 to the 8 ions per second, which is really fast because that's 10 to the 5th times faster than the most efficient carrier active proteins transporters. And channel proteins are used extensively in nerve cells, in which they are responsible for transduction of electrical signals. So it's also called as action potentials. The stimulus causing channel proteins to open or close can have a variety of forms. It can be change of voltage across the membrane for the voltage-gated channel. It can be ligand binding for ligand-gated channels. In this case, ligand may come from either the extracellular environment or from other cells, or intracellular environment too. And also, the stimulus can be mechanical stress for mechanically gated or stress-gated channels. And there has been more than 100 types of ion channels that are discovered. And the common ion channels are those responsible for the potassium transport, especially in neurons. And in particular, some of these are open continuously all the time, so-called potassium ion leak channels, which is critical for maintaining the voltage potential across the cell membrane. And sodium-potassium pump maintains osmotic pressure by keeping the potassium ion concentration low at the expense of increasing the number of potassium ions inside of the cell. So although you're getting 2 potassium ions inside of the cell, you're pushing out the 3 sodium ions. So overall, you're having a less number of the ions or the molecules inside of the cell, which can help them maintaining the low osmotic pressure. And this is required to balance the anion cation concentrations in the cell, where the most organic molecules in the cytosol are cations. And the concentration of potassium ions is kept in equilibrium by the potassium ion leak channels. Let's discuss more about this potassium leak channel, which is quite interesting. For many years, it was a puzzle how this potassium leak channel functions, which has a high specificity only for the potassium ions. So it definitely can distinguish between sodium and potassium ions, which are both small spherical positively charged ions. And when you look at the radius of these ions, they are quite similar as 0.09 nanometer for sodium ion and 0.133 nanometer for potassium ions. But besides, the sodium ion is even smaller, so if it were to be just a channel that passes through all the ions, then sodium, which is a smaller ion, should pass through pretty easily. But that's not the case, so that means the channel does not select for size. And also, there's a fast passage for the potassium ions too, which indicates that the channel does not have specific binding sites for potassium ion either. Finally, the puzzle was solved with the determination of the structure using the bacterial potassium channel via X-ray crystallography. Potassium ion channel have 4 protein subunits, although only 2 are shown in this picture. And at the cytosolic end of the channel, these subunits have negatively charged amino acids which attract cations such as potassium and sodium ions. And carbonyl oxygens line the interior of the pore here with a precise separation, and that provides a key feature for the selectivity of these potassium channels. And the hydrated ions, which is potassium or sodium ions, that's surrounded by water molecules for the electronegativity of these oxygen atoms on the water molecules interacting with the positively charged ions. These ions can only proceed through the pore by changing their coordination with water molecules, coordinating with these carbonyl oxygens in the channel. So it'll get into the pore inside of the potassium channel, now interacting with these oxygen molecules. And only potassium is able to do this efficiently since the diameter of the pore has been tuned for this potassium ion. The sodium ion is too small, so it's unable to make enough coordination to make the transport energetically efficient. And key to the ion selectivity of the potassium channel is an ion filter, which consists of these carbonyl oxygens that are aligned nicely in the pore of the channel. And this earned the Nobel Prize in 2003. So Roderick Buchanan published in April 1998 the first crystal structure of an ion channel. And that actually showed and explained this ion filter with the selectivity for potassium ions over the sodium ion. The protein secretory pathway explains the process for the synthesis of an integral protein. So in the rough ER, or rough endoplasmic reticulum, where the translation happens, the SRP receptor and the translocon interact with the SRP, which recruits the ribosome with the signal peptide attached there, and the translation is initiated, and the signal peptide is removed right after that, and the translation starts from the N-terminal region, and then as soon as the nascent polypeptides are generated from the ribosomes, the folding and the glycosylations happen at the same time, and once all the translation is done, the transmembrane domain is naturally stuck on the rough ER membrane to become the integral protein already. After the translation of the integral protein on the rough ER, these integral proteins are embedded on the vesicles and that are coming from the ER that travels to Golgi apparatus. And then on the Golgi apparatus, those integral proteins are further processed for their post-translational modification, for their maturation, functionality, as well as localization. So finally, they're going to move to the plasma membrane or some other organelles, including lysosomes too.